Hi, my name is Dalia Al-Gamdi. I am a PhD candidate at the University of British Columbia and I work under William Shaw supervision. My PhD project focuses on building controlled vocabulary as an application ontology for next generation biobanks. The biomedical domain is known to be interconnected. It includes different data types such as lifestyles and behaviors, medical history, genetic data, demographic data, and laboratory measures. Like in many other domains, a growing number of data resources increase the level of data diversity, especially when new revolutions unfold, such as the omics studies, along with new softwares and hardwares created continuously. Nowadays, managing biomedical data has been subject of various research projects tackling two main challenges, which are data being incompatible and therefore difficult to impossible to query. Over the last few years, controlled vocabulary ontologies proved their ability to overcome these challenges, and nowadays, ontologies are well established in the biomedical area. By using ontologies to annotate data from multiple resources, we will be able to map concepts in a particular database with the same concept in another one. Here, for example, the European Nucleotide Archive used the term instrument to label the name of the company that produced sequencer equipment along with the model number. On the other hand, the National Center for Biotechnology Information used the term sequencing technology to label the same concept. For large datasets like biobanking data, different labeling and different usage of terms will make it challenging for humans and impossible for computers to integrate data. Nowadays, biobanks and public research platforms such as CINECA consist of both biological samples and data associated with them, and that include huge digital data that are produced by high-throughput technologies. In our lab, we were interested in the integration of omics metadata standard into the biobanking domain ontology. So we built next generation biobanking ontology, which is an open source ontology built mainly for the integration of omics metadata into the current available resources of biobanking domain ontology. The knowledge of the ontology was acquired from multiple resources, including the College of American Pathologists requirement and checklist. So CAP provides accreditation to medical laboratories through its Quality Solution Program, and this quality program includes a set of standards and requirements for data reporting, and NGBO was built based on those reporting requirements. Other resources include molecular pathology clinical reports, laboratory and biobank standard operating procedures, and to not reinvent the wheels, whenever available, terms from related domain ontologies in OpenFoundry were used. NGBO is composed of a term hierarchy that describe analytical workbench and bioinformatics processes and related metadata that are required to be reported, including terms describe processes such as genetic material isolation and different bioinformatics analysis, material and objects such as kits and instruments, data and data descriptions such as different genomics data types and their formats, and recursive methods and protocols used for different assays. By mapping the variables and attributes from each cohort or database to an ontology model, we are generating a data in a format called RDF triples. An unlimited number of RDF triple can be uploaded into single graph database, and then queries can be performed over the graph. I will show a brief example of RDF triple in next slide. However, if you are new or need more understanding about ontologies and how to use them to annotate data, please refer to Dr. Emma Griff's videos on Pono2Po page. Here is an example for mapping a free text from clinical report to ontology terms in an RDF format. The text includes information about target region enrichment platform and the DNA sequencer used in sequencing assay. RDF triple is a way of describing information. It is made up from 3 parts. The first part is subject. So for example, alumina high-seq is our subject here. The second part is predicate. In this case, it's the ISA relationship. And the third part of the triple is the object, which is in this case the DNA sequencer. This graph contains multiple triples, and unlimited number of RDF triples can be added into it. And it could be expanded infinitely with more and more pieces of information. An example of a use case, a researcher is looking for a biobank which include colon samples and their associated RNA sequence data. Here, we use the description logic query to query a synthetic data attributes mapped to NGBO terms. You can have a closer look at NGBO in my GitHub page. Thank you very much for watching this video. Please leave your comments or questions.